Welcome to the deep dive. Today we're tackling something truly chilling. Mass methanol poisoning.
It's a recurring tragedy, isn't it?
Absolutely.
Yeah.
We've seen devastating outbreaks, like the big one in the Czech Republic back in 2012, and more recently, really awful cases in Iran and Rwanda.
And the root cause is almost always the same. People consuming alcohol well, it's not standard, it's illicit, sometimes industrial grade or just dangerously adulterated.
It's horrifying. Permanent blindness, organ failure.
Yeah.
The death toll can be incredibly high. And it all starts with a simple chemical confusion.
Exactly. People hear methanol and they think poison, which is true, eventually. But the immediate killer, the thing that actually causes the damage, isn't the methanol itself. It's what happens after you drink it.
Right. So you've got ethanol, that's the alcohol in regular drinks, and then you've got methanol, the dangerous one. But you're saying the initial molecule isn't the weapon.
Not directly, no. The body has this enzyme, alcohol dehydrogenase ADH. Its job is to break down alcohol. It sees methanol and thinks, okay, I know what to do with this. And it starts the process. First step, formaldehyde.
Which is not good. Embalming fluid, right?
Exactly. Highly toxic, very reactive. But it doesn't stop there. The body quickly converts that formaldehyde into the real villain, formic acid.
Formic acid.
That's the chemical that does the damage. It goes straight for the mitochondria, the power plants inside our cells.
And shuts them down.
Shuts them down hard. Causes this profound systemic acidosis, basically makes the blood dangerously acidic and damages tissues all over the body.
Okay, so that sets up our mission for this deep dive. We need to unpack this whole stack of sources and look at three key areas. First up, where does this methanol actually come from? Naturally versus, the criminal side.
Right. Then second, we'll get into the technology. Some really amazing new tools are being developed to spot this stuff fast. Handheld devices, AI, really cutting edge.
We definitely need that speed.
Yeah.
And finally,
we have to understand the biological damage. What exactly does formic acid do? What changes in the body? What are the signs in the brain, the eyes? And critically, what tells doctors if someone's likely to survive?
understanding the science here is absolutely key to fighting this. Let's get into it. All right, part one, the origin story. Because methanol isn't just some industrial nasty, is it? It's actually around naturally.
It really is. It's kind of surprising. You find low levels in the air we breathe, especially if you're, say, walking through woods or near a freshly cut lawn. Plants give it off.
Okay. But how does it get into drinks, even the legitimate ones?
The main natural source in our diet is something called pectin.
Pectin, like the stuff you use to make jam set.
That's the one. It's a type of fiber found in the cell walls of fruits and vegetables. Apples, plums, berries, lots of pectin.
And methanol is hiding in there.
Well, it's chemically bound within the pectin structure, a methyl group specifically. And this gets released through a process called demethylation.
Demethylation, taking the methyl group off.
Exactly. And there's an enzyme that speeds this up, particularly as fruit ripens. It's called pectin methylesterase, or PME. So ripening fruit naturally releases a bit of methanol. It's part of decomposition.
So if we eat fruit, we're consuming pectin, and our body handles that. We make a little methanol inside us.
We do, yeah. It's an endogenous source. And our gut bacteria play a big role, too.
Ah, the microflora connection.
Right. Humans don't really have the enzymes to break down pectin fully, but some of our gut microbes do. They have that PME gene, so they ferment the pectin, and a byproduct is, again, a small amount of methanol.
So our body is constantly dealing with tiny amounts of this stuff.
Constantly producing and clearing small, manageable amounts. And the metabolites, like formaldehyde, too. It's all tightly regulated, usually completely harmless at these low levels.
Okay, that makes sense then why some legal alcoholic drinks can have naturally higher methanol levels.
It absolutely dictates the regulations. If you're distilling spirits from things really rich in pectin, think cherries, plums, maybe some grapes, you're going to concentrate that naturally occurring methanol along with the ethanol.
How high can it get naturally?
The sources mention some studies finding levels in, say, raw fruit distillates, ranging from maybe 18 up to 25 grams of methanol per liter of pure ethanol.
Whoa, 25 grams per liter sounds incredibly dangerous.
It would be, but that's in the raw spirit before any blending or regulation. Commercial spirits are subject to very strict limits. That's the key.
So the regulations bring it way down.
Way down. In the U.S., for fruit spirits, the limit is closer to 6 or 7 grams per liter of pure ethanol equivalent. The EU has varying limits depending on the type of spirit, but they're all designed for safety, much lower than that natural maximum potential, maybe around 1,500 grams per hectoliter of pure alcohol.
So the system works for legal regulated drinks, but it completely falls apart with the illegal stuff.
Precisely. Our bodies handle the tiny natural background levels, and regulators handle the higher levels in specific fruit spirits. The real danger, the lethal danger, explodes when we get into unrecorded and illicit alcohol.
Which brings us right to the heart of the problem, the global threat of these dangerous spirits. ##B, the threat of unrecorded and illicit spirits.
And this is where it gets really scary. These deadly high concentrations of methanol, they're almost exclusively found in what's called unrecorded alcohol.
Unrecorded meaning homemade, smuggled.
Yeah, homemade, bootlegged, illegally produced stuff with zero quality control. It's a huge problem in places like Eastern Europe, South America, parts of Africa.
Is this just a small fringe market?
Not at all. Take Turkey, for instance.
Yeah.
The sources suggest something like 30%, nearly a third of all alcohol consumed there is unrecorded.
30%? That's massive.
Yeah.
That's a huge unregulated supply chain.
It's a public health time bomb. And when you look at the chemical analysis from actual poisoning outbreaks, it's terrifying.
Let's look at those case studies. Rwanda and Iran really highlight the chaos, right?
They really do. In Rwanda, they analyzed some of these illegal drinks. The ethanol content was all over the place, from barely alcoholic, like 3.8%, up to almost pure alcohol, 98.9%.
Okay, wildly inconsistent.
Huh.
But the methanol,
That's the shocking part. Ethanol levels range from 32% up to an unbelievable 58.3% by volume.
58%, that's not a beverage, that's chemical waste. The legal limit is usually under 0.5%, right?
Exactly, often much lower. And there was another crucial finding in those Rwandan samples.
What was that?
None of them managed to be dangerous in both ways at once. They were either way too high in ethanol or they were packed with methanol, not both.
What does that suggest? Just bad distilling.
It suggests more than that. Probably not just poorly controlled fermentation. It points towards deliberate adulteration, maybe mixing in industrial solvents or using denatured alcohol, or just straight up adding methanol because it's cheap.
Okay. And the situation in Iran with those homemade raisin spirits.
Similar story, sadly. Very wide range of methanol content. The average was over 7,500 milligrams per liter, but the maximum they found was over 12,000 mg/L lethal levels.
And is there a way to tell chemically if it's deliberate contamination versus just, bad homebrewing?
There often is, yes. A key clue is the absence of other types of alcohol.
Other types.
Things called higher alcohols or fusel oils. These are natural byproducts when yeast ferments sugars from agricultural sources, grains, fruits, molasses, that kind of thing. They have a distinct chemical signature.
Okay, so proper fermentation leaves behind these fusel oils.
Right. So if you find a spirit that's got tons of methanol but almost no fusel oils, that's a huge red flag.
What does it mean?
It strongly suggests the makel didn't start with agricultural ethanol at all. They probably used cheap industrial ethanol, maybe stuff intended as solvent, or even worse, just added methanol directly to stretch the volume or mimic the kick of ethanol. That lack of higher alcohols alongside high methanol is practically a smoking gun for dangerous illicit production.
Got it. The missing pieces of the natural chemical fingerprint give the game away. Now, this whole issue of contamination in drinks, it's not just limited to alcohol, is it? The sources brought up a really interesting parallel with non-alcoholic drinks and packaging.
Yes, it's a great point about broader consumer risk. They looked at endocrine-disrupting chemicals, EDCs, in everyday packaged beverages.
EDCs, those are chemicals that mess with your hormones, right?
Exactly. Things like BPA, bisphenol A, which used to be common in plastics and can linings. And the parallel is strong. Whether it's methanol secretly added to spirits or EDCs leaching from a can, the underlying issue is a breakdown in quality control and regulation, leaving the consumer exposed.
What did they find specifically with BPA in drinks?
They found surprisingly high levels, especially in things like canned coconut water imported from Southeast Asia. Places like Vietnam and Thailand were mentioned.
So stuff on regular supermarket shelves.
Potentially, yeah. It highlights that the risk isn't just from some back alley still, but can also enter the legitimate global supply chain if controls aren't tight enough.
And the exposure levels weren't trivial, were they?
Far from it.
Yeah.
For BPA, in the worst-case scenarios they calculated, the exposure could be 2,000 times higher than the accepted safety threshold for non-alcoholic drinks.
2,000 times. That's staggering.
It really is. And it underlines how vulnerable consumers are, especially kids who might drink a lot of canned or packaged beverages. The fundamental point holds. Inadequate regulation or control, whether for illicit booze or imported coconut water, creates serious hidden chemical dangers.
Which makes the need for rapid detection absolutely critical. We can't wait for lab results if the danger is potentially that high and that immediate.
Exactly, which leads us perfectly into the next part, the race to develop faster, better detection tools.
Okay, part 2. If methanol poisoning is this race against time because of that formic acid buildup, then knowing instantly if a drink is contaminated is crucial. This means moving beyond the old lab methods, #tag the limits of traditional methods.
That's the bottleneck they're trying to break. The traditional gold standard method is gas chromatography, GC.
Which is very accurate.
Extremely accurate, yes, but it's slow. It needs bulky, expensive equipment, usually requires sample prep like distillation, and you need highly trained people to run it and interpret the results.
Not exactly portable or fast.
Not at all. And other methods, like redox titration, while accurate, also take time. In a mass poisoning situation, maybe dozens of people showing up at a hospital, you just don't have hours to wait for a GC result for each person or each suspect bottle.
Lives depend on speed. So what's emerging? Let's talk about these new portable rapid technologies. That handheld sensor sounds amazing. #B. The rise of portable and rapid screening technologies.
Yeah, the ABEG device described in the sources sounds like something out of science fiction almost. It's a pocket-sized analyzer. Think of it as a highly specialized electronic nose.
An electronic nose. How does it work?
It uses a special film, a chemoresistive sensing film. Basically, its electrical resistance changes when certain gas molecules stick to it.
What's the film made of?
It's quite sophisticated. They use tin oxide nanoparticles, SNO2, which is a common sensor material, but they dope it with palladium PD-doped SNO2.
Why the palladium?
Palladium acts as a catalyst. It makes the sensor much more sensitive and selective, particularly for alcohol molecules like ethanol and methanol.
Okay, so it sniffs the air or the vapor from the drink.
Exactly. And to help it tell the difference between ethanol and methanol, which are chemically quite similar, they pair the sensor with a tiny separation column, something called 10x-TA.
So the column gives the molecules a bit of a head start difference before hitting the sensor.
That's the idea. It helps separate them slightly in time. And the speed is incredible. The total analysis, time sample prep, running the test, getting the result is under 90 seconds.
90 seconds. That's faster than making instant noodles.
Pretty much. And they tested it on real drinks beer, high-proof spirits like Iraq and Stroh rum, and it worked.
How do you see the results?
It sends the data wirelessly to a smartphone app. So immediate visualization right there in the field. It's a huge step towards decentralized, rapid screening.
But you mentioned SNO2 sensors sometimes struggle to tell methanol and ethanol apart. How did another approach tackle that fundamental selectivity problem?
Right, that's always been the challenge. This leads to the Tonazei research, which combined clever sensor design with machine learning.
Okay, AI gets involved.
Exactly. They realized that just having a better sensor material might not be enough, so they took a single SNO2 nanowire sensor.
Nanowires, okay.
But instead of just measuring its response at one temperature, they used a thermal gradient. They measured the sensor's electrical response at 5 different temperatures.
5 temperatures. Why does that help? Is it like sniffing something hot versus cold?
That's a great analogy, yes. Methanol and ethanol interact with the sensor surface slightly differently depending on the temperature. How strongly they stick, how quickly they react, it all changes with heat.
So measuring at 5 different temperatures gives you 5 slightly different views of the same vapor.
Precisely. It creates a much richer data signature, like a complex fingerprint for each alcohol, and especially for mixtures. It's much harder to confuse methanol and ethanol when you have this multi-temperature profile.
And that's the data you feed to the AI.
That's the data you feed to the machine learning algorithms. They mention using things like PCA, LDA, and SVM, basically powerful pattern recognition tools.
Principal component analysis, linear discriminant analysis, support vector machine. Fancy names for teaching the computer to spot the methanol fingerprint.
Essentially, yes.
Yeah.
The computer learns to distinguish the subtle patterns in that thermal gradient data that correspond to pure methanol, pure ethanol, or specific mixtures.
And how well did this AI-powered nose work?
The results were stunning. They achieved 100% accurate classification between methanol and ethanol.
100%, even in tricky mixtures.
Yes, even in complex alcoholic samples and under varying humidity, which can often mess up sensors. And the error in quantifying the amount was tiny, only about 3 parts per million on average.
Wow, and how fast?
Result in about 2 to 3 minutes, so slightly longer than the AB, but still incredibly fast and with apparently perfect accuracy in telling them apart. It's a massive leap. #C spectroscopic shortcuts. BOYACI, grew or day.
Okay, so sensor arrays with AI are one route. What about other approaches? Spectroscopy comes up using light to identify chemicals. Are those getting faster too?
They are. While spectroscopy often needs larger gear than those handheld sensors, the advantage is that some techniques can analyze the sample directly without needing that time-consuming chemical separation step you have in GC.
Like shining a light through the drink.
Kind of. One method mentioned is Raman spectroscopy. You shine a laser on the sample and you look at how the light scatters back.
And different molecules scatter light differently.
In a unique way, yes.
Yeah.
Based on their molecular vibrations, every molecule has a characteristic Raman spectrum, like a fingerprint.
How fast is that?
Very fast analysis in less than 30 seconds, according to the source. And it's non-destructive. You just shine the laser, read the spectrum. By comparing it to known standards, they could accurately quantify methanol directly in spirits, no distillation needed.
30 seconds is impressive. Then there was another, more unusual spectroscopic method mentioned. Something involving plasma.
Ah, yes, the SAGI-MES. That stands for Solution Atmospheric Glow Discharge Molecular Emission Spectrometry.
Okay, say that 5 times fast. What does it do?
It's pretty wild. You take the vapor from the alcohol sample and inject it into a tiny, super hot cloud of energized gas, a microplasma.
Like creating a miniature star.
Sort of. The intense energy in the plasma breaks the alcohol molecules apart, and then you look at the light emitted by the fragments, specifically carbon-containing fragments like carbon monoxide molecules.
So you're not seeing the methanol itself, but the light from its broken pieces.
Exactly. The pattern of light emission tells you what was there. The big breakthrough, they reported, was figuring out how to get way more of the alcohol vapor into the plasma, a 6-fold enrichment.
Which makes the signal much stronger.
Much stronger, much easier to detect and quantify accurately. They compared it to the traditional titration method and found it was very accurate, with errors only between about -1.7% and +7.0%. Another promising rapid technique. #D low-cost field methods. KO to daddy.
These high-tech sensors and spectrometers sound amazing, but probably expensive. What about places that don't have the resources for that kind of equipment? Are there simpler, cheaper field tests?
That's a crucial point, and yes, there are efforts there too. One source described a method based on good old-fashioned colorimetry detecting color changes.
Like a pH strip, but for methanol.
Sort of, but a bit more involved. It still requires a couple of steps, but using simple equipment. First, they use microdistillation, a small-scale distillation, just to separate the methanol vapor from the rest of the liquid.
Okay, so you still need to isolate it somewhat.
Yes, to avoid interference. Then they use a chemical reaction, adding acidic permanganate solution to convert that methanol into formaldehyde.
Back to formaldehyde again.
Right. And then they add another chemical, chromatographic acid. This reacts with the formaldehyde to produce a colored compound.
And the more methanol you started with, the more formaldehyde you make, the deeper the color.
Exactly. You can then visually compare the color to a chart or use a simple colorimeter device to measure the intensity, which tells you the concentration.
How sensitive is that?
The detection limit mentioned was 183 milligrams per liter, so maybe not as sensitive as the AI sensor, but potentially good enough to flag dangerously contaminated samples.
And the big advantage is cost and simplicity.
Absolutely. It's designed to be inexpensive and usable in settings without advanced labs or highly trained staff. It's a vital tool for resource-limited areas facing this threat.
Okay, we've seen how methanol gets into drinks and how we can detect it. Now let's follow it into the body, part 3. The damage it causes. This all comes down to that formic acid metabolite, #GCA, the toxic pathway and antidotes.
It really does. So methanol gets absorbed, goes to the liver, enzyme ADH1B converts it to formaldehyde, which is bad.
But quickly becomes formic acid.
Which is worse. That formic acid circulates and targets the mitochondria. As we said, it blocks mitochondrial respiration, specifically an enzyme called cytochrome-seq oxidase, right at the end of the energy production chain.
So the cells can't use oxygen properly to make energy, ATP.
Correct. It leads to cellular asphyxiation, massive lactic acid buildup from cells trying to make energy without oxygen, and that severe metabolic acidosis. This is why it hits organs with high energy, demands hardest of the brain, the optic nerve, the kidneys, the heart.
Which explains the devastating symptoms. Now, the antidote seems counterintuitive. You give someone more alcohol, specifically ethanol. How does that work?
It's all about competitive inhibition. Think of that ADH1B enzyme in the liver as having a limited capacity, like a busy checkout counter.
Okay.
Both methanol and ethanol want to use that counter, but the enzyme has a much, much stronger preference for ethanol, maybe 10 to 20 times stronger.
So if you flood the system with ethanol,
You basically keep the enzyme busy processing the ethanol. It physically blocks the methanol from getting access to the enzyme.
so you stop, or at least drastically slow down, the conversion of methanol into that toxic formic acid.
Exactly. It buys time. It doesn't get rid of the methanol, but it stops the production of the really dangerous stuff, allowing the unchanged methanol to be cleared from the body, usually through dialysis.
Is ethanol the only option?
There's also a drug called foncoazole, which does the same thing, blocks ADH1B, and is often preferred in hospitals as it's easier to dose consistently. And they also give folinic acid.
Why folinic acid?
That helps the body break down the formic acid that has already been produced. So the treatment strategy is twofold. Block new formic acid production with ethanol, or fermazole, and speed up the removal of existing formic acid with folinic acid and dialysis.
You mentioned dosing is critical, and something about zero-order kinetics for ethanol.
Right, this is important for understanding the treatment. Once you drink a relatively small amount of ethanol, maybe just 30 milliliters or so, the ADH enzyme system in the liver gets completely saturated. It's working at its maximum speed.
Okay. So it can't work any faster, even if there's more ethanol.
Correct. That means the body eliminates ethanol at a constant rate, like so many grams per hour, regardless of how high the blood alcohol concentration is. That's zero-order kinetics. It's different from most drugs, which follow first-order kinetics. Elimination rate depends on concentration.
Why does that matter for the antidote?
It means you need to give a continuous infusion of ethanol to maintain a constant therapeutic blood level that keeps that ADH enzyme blocked. You can't just give one big dose. You have to keep the block active until the methanol is gone.
Let's zoom out from the cellular level. When someone is acutely poisoned, what does the systemic chaos look like? The sources mentioned proteomic studies from the Czech outbreak. What proteins change?
Proteomics gives you this amazing snapshot of all the proteins changing in the blood. And yeah, the Czech study comparing acutely poisoned patients, long-term survivors, and healthy controls showed massive shifts across multiple systems. Blood coagulation was one major area.
Clotting goes haywire.
Pretty much. They saw significant increases in things like von Willebrand factor, involved in platelet adhesion, carboxypeptidase N, and alpha-2 antiplasmin, which inhibits clot breakdown. But at the same time, other key factors like kininogen 1 and plasminogen needed for clot breakdown were decreased.
So the body's trying to clot and stop clotting all at once.
It suggests a highly dysregulated state, yeah,
Right.
potentially leading to both thrombosis, unwanted clots, and bleeding risks. It's biochemical chaos. Now, the researchers did note that giving heparin as part of the treatment might have influenced some of these coagulation markers.
Okay. What about the immune system? Is there a big inflammatory response?
Definitely. Key markers of the immune and inflammatory response shot up. This included proteins like complement factor I, and particularly complement factors C3 and C5.
C3 and C5 are part of the complement cascade, right? A first-line defense.
Exactly. Seeing them elevated indicates a massive activation of this system, likely triggered by the widespread tissue damage. CD14, a marker on monocytes, a type of white blood cell, was also increased, indicating immune cell activation.
Is that purely a sign of damage, or is the immune system trying to help?
It's mostly a sign of damage, but there's some thought that maybe activating C3 and C5 could have some neuroprotective effects in certain contexts. It's complex. The body is clearly in crisis mode.
Any other major protein changes? Metabolism?
Yeah.
Transport?
Yes. Indicators of metabolic stress are up too. They found increased retinol-binding protein, which carries vitamin A, and also increased apolipoprotein AI and A2, which are crucial for transporting cholesterol and lipids.
So even fat transport is disrupted.
It seems like it touches almost everything. These protein changes paint a picture of the body scrambling to cope with profound cellular energy failure and widespread damage, affecting clotting, immunity, and basic metabolic processes. #, see neurological and ocular sequelae.
For those who survive the acute phase, the damage often isn't over. The long-term consequences, especially neurological and visual, can be devastating. Formic acid seems to target specific areas. What do MRI scans show in survivors' brains?
The MRI findings are quite characteristic, almost a signature of methanol poisoning. The most common finding, seen in over 3/4 of cases in one study, 77.6%, is damage to the putamen.
The putamen. It's deep in the brain, part of the basal ganglia,
Yeah,
involved in movement.
exactly. And typically the damage is bilateral, affecting the putamen on both sides of the brain. This bilateral putamen necrosis is highly suggestive of methanol toxicity.
Does the pattern of damage matter?
It seems so. The sources mentioned that if the necrosis, the tissue death, was asymmetrical, worse on one side than the other, it tended to correlate with more severe long-term neurological problems.
And this pattern helps doctors distinguish it from other brain injuries.
Yes, that's clinically very useful. For example, carbon monoxide poisoning can also cause damage to the basal ganglia, but it more typically affects a different structure called the caudate nucleus. Methanol poisoning characteristically hits the putamen.
What about the eyes? Vision loss is tragically common.
Yes, the MRIs often also show signs of damage to the optic nerves, things like nerve enhancement or, later on, atrophy shrinkage. This was seen in over half the cases, 56.9%. The combination of bilateral putamen necrosis and optic nerve damage is a very strong indicator for methanol poisoning.
Can they track the eye damage more closely over time?
Yes, using advanced imaging like optical coherence tomography, angiography, or OCTA, this lets them look at the structure of the retina and its tiny blood vessels in incredible detail.
What did the OCTA follow-up studies show?
They showed progressive damage. Over a period of just 3 months after the poisoning, survivors had a measurable thinning of the inner layers of the retina and, critically, thinning of the retinal nerve fiber layer, our NFL.
The RNFL, that's the layer containing the axons traveling from the eye to the brain, right?
Exactly. Losing those nerve fibers is what causes the vision loss. They also saw reduced density in the tiny blood vessels near the center of the retina, the perifoveal region.
And does this thinning show up physically?
Yes. It leads to something called optic disc cupping. As the nerve fibers die off, the optic disc, where the nerve leaves the eye, starts to look hollowed out or cupped. The OCTA studies showed this cupping increased significantly over time, mirroring the decrease in RNFL thickness.
Why is the optic nerve so vulnerable? What's the mechanism?
It comes back to those mitochondria and energy supply. The formic acid seems particularly toxic to the support cells within the optic nerve head specifically, prelaminar astrocytic glial cells, and oligodendrocytes.
These are the cells that nourish and insulate the nerve axons.
Correct. When their mitochondria are poisoned by formic acid, these crucial support cells die. And when the support cells die, the nerve axons they maintain also die. That axon loss is permanent, leading to irreversible blindness. #D, prognostic indicators for mortality.
This is incredibly grim. When a patient comes into the ER, doctors need to know quickly how likely they are to survive. What are the biggest warning signs, the strongest predictors of a poor outcome?
Clinically, some signs are fairly obvious indicators of severe poisoning. Being in a coma or having significant respiratory depression, not breathing well. Those are immediate red flags.
What about lab tests?
Metabolic markers are huge. One study highlighted hyperglycemia, high blood sugar on admission, as a bad sign. The median glucose they reported was quite high, around 185 milli-UDL, suggesting a major stress response or metabolic breakdown.
But the single most critical chemical indicator, is it the acidosis?
Absolutely. Severe metabolic acidosis, that dangerously low blood pH caused by the formic acid, is probably the single strongest predictor of death. After adjusting for other factors, one analysis found that severe acidosis increased the odds of dying by almost 4 times. Odds ratio 3.79.
So correcting that acidosis immediately is life-saving.
It's a top priority, along with starting the antidote and dialysis. If the pH is critically low, the prognosis is very poor unless aggressive treatment can reverse it quickly.
Any other lab results that help predict severity or the need for treatment like dialysis?
Yes, some other findings correlated with a tougher course. Things like low core body temperature, low levels of BUN, blood urea nitrogen, and creatinine, which usually indicate kidney function, but might be low initially before kidney failure sets in, were associated with needing more sessions of hemodialysis.
Interesting. Kidneys are working hard to clear it, perhaps.
or maybe reflecting a different aspect of the metabolic disturbance. Low lactate dehydrogenase, LDH levels, were also noted in some comparisons, although high LDH usually indicates tissue damage, so the reason for low levels being significant might be complex. These markers help guide how aggressive the detoxification needs to be.
And finally, thinking about toxicology after death, or even just how it distributes. Does methanol hang around? Where does it concentrate?
It does persist. Methanol and formic acid can be found in body fluids and tissues urine, the vitreous humor of the eye, liver, brain for days after death, which can be important for postmortem diagnosis.
Does it spread out evenly like regular alcohol?
Not quite. The distribution pattern is different from ethanol. Ethanol distributes widely in body water. Methanol, however, showed higher partition coefficients in tissues, especially the liver, compared to body fluids like urine.
Higher concentration in the liver. Why?
It likely reflects where it's being metabolized. Since the liver is the main site where ADH converts methanol to formaldehyde and formic acid, the methanol might linger there more, bound up in that enzymatic process, leading to higher tissue concentrations compared to fluids. It's another distinguishing feature compared to ethanol.
Okay, we've covered a lot of ground, from pectin in fruit to AI-powered sensors to mitochondrial failure. Let's try and crystallize the main takeaways from this deep dive.
I think one of the biggest shifts is definitely in the detection technology. We're moving away from slow lab-bound methods like GC
Towards these rapid, portable solutions.
Exactly. Things like the ABJ handheld sensor or the TONA-ZE approach using SO2 nanowires, thermal gradients, and machine learning. They offer near-instant, highly accurate identification of methanol, even distinguishing it perfectly from ethanol in minutes.
That speed is revolutionary for responding to outbreaks or screening suspicious products.
Absolutely. And on the biological side, we have a much clearer picture of why methanol is so devastating. It's the formic acid metabolite targeting mitochondria.
Leading to that specific pattern of damage.
Right. The systemic chaos showing up in the proteome, affecting coagulation, immunity, metabolism. And then the hallmark neurological damage, that bilateral putamen necrosis in the brain, and the progressive thinning of the retinal nerve fiber layer causing blindness. Knowing these specific targets is crucial for understanding the pathology.
And that understanding directly informs treatment and prognosis.
It does. Recognizing the critical importance of severe metabolic acidosis as a predictor of death emphasizes the urgency of treatment blocking methanol metabolism with ethanol or fombisol, clearing methanol and formic acid with dialysis, and supporting the patient's physiology. Rapid diagnosis linked to prompt, targeted treatment is the key.
Now, stepping back for a final thought, we talked about methanol being naturally present, and our bodies having systems to deal with tiny amounts, even metabolizing it to formaldehyde.
Yes, the fact that we have specific enzymes like formaldehyde dehydrogenase FDH, also known as ADH3, dedicated to handling even low endogenous levels of formaldehyde is fascinating. It suggests formaldehyde might play some biological role, or at least that detoxification is essential.
So if our body carefully manages tiny internal amounts of this stuff, and we know formaldehyde is reactive and potentially linked to neurological issues, what about chronic exposure?
That's the lingering question, isn't it? We focus rightly on the horror of acute mass poisonings. But what about the millions of people worldwide who regularly consume unrecorded alcohol over many years? They might not be getting acutely lethal doses, but they could be getting chronically elevated low-level exposure to methanol and its toxic metabolites.
What could the long-term subtle health effects of that kind of exposure be? Especially if formaldehyde itself might play a role in other neurological disorders.
Exactly. Beyond the immediate crisis of poisoning, the potential long-term impact of chronic, low-grade methanol and formaldehyde exposure from unregulated alcohol consumption is a huge, largely unanswered public health question. That's something we really need more research on.